All right, so here we have a cell surrounded by a cell membrane, and here's the nucleus and the nucleolus. And remember, the nucleolus makes these things called ribosomes, and here we see ribosomes floating throughout the cell in the cytosol. And we also see ribosomes attached to what we call the rough ER, rough endoplasmic reticulum. The ER is a series of tunnels and passages that are attached to the nucleus. If there are ribosomes embedded in the wall of the ER, it's rough ER. Compared over here to the smooth ER, there are no ribosomes. And again, both ERs are connected to the nucleus. Inside the nucleus is the DNA, which is the instructions for how to make stuff. So basically what happens is we make a copy of the directions, and we'll learn about that in a couple weeks. But we take these directions here. This is the master copy, the instructions. We make a copy of it. The copy goes here through the rough ER to one of the ribosomes, and the ribosomes take those instructions and make things. And what they make, we package in these little containers called vesicles. So here's a vesicle, and it moves over here. It's basically like a food tray, like a cafeteria tray. And it comes over here, and the vesicle merges. It's almost like two soap bubbles coming together, merges with this stack of membranes, and they're called Golgi Bodies or Golgi apparatus. And the Golgi apparatus has a couple jobs. And basically what the Golgi do is they check to make sure the contents of the ribosomes, the contents of the vesicles rather, are well made. And if they are well made, they will then repackage them and put an address on them, just like the post office, and put them into another vesicle. And that vesicle is off to different parts of the cell, or can even leave the cell. The problem occurs is if the ribosomes over here put product in the vesicle, which came over here, and let's say that product is not made well. So what the Golgi does is it targets it for destruction. So the Golgi Bodies puts that product into a vesicle, but in this case, the vesicle comes over here and binds with another organelle called a lysosome. And what lysosomes are is they're organelles that contain digestive juices, digestive liquids inside them, enzymes. And what these enzymes do is they will then chop up what the vesicle brought and spill up the contents back out into the cell to recycle. So it's a really efficient means of checking products and recycling. And then lastly over here we've got the mitochondria. And what the mitochondria does is it takes the energy stored in sugar and other substances and breaks those substances apart and releases the energy. Because everything that we're doing here requires energy. And we'll be learning about that in more detail in a few weeks.